So today we are going to discuss diversity of immunoglobulin synthesis that refers to diversity of the adaptive immune response. इसका मतलब क्या है? How many types of antibodies our body can synthesize? 10 raised to the power 11. So that is too large a number to be counted rather. So we can say that limitless immunoglobulins are synthesized. The reason is very simple because limitless is the number of antigens, the biomolecules or the other molecules to which our body is exposed. And each immunoglobulin has got a different specificity. Each immunoglobulin is only for against a particular antigen only. So this is called specificity. So the two terms, they appear antithetical, but they complement each other. Limitless is the number of immunoglobulins synthesized. Each is specific for a given antigen only. But the genomic material is limited. 10 raised to the power 9 base pair. तो यहाँ पर क्वेश्चन यही आता है कि do we have enough genetic material to synthesize, by most conservative estimate, 1 million different types of immunoglobulin, 10 raised to the power 6. Do we have enough genomic material? So the question I'm reading out, how can genes for some million antibodies find space in human genome? Of course, human genome is gigantic, very, very large, 3000 mega base pairs. But all of it is not coding for protein. As you can see in this figure, only 1.3% is protein encoding. Rest all introns, tandem repeats, intersperse repeats, etc. They are all non-coding. They do not code for any protein. So ये 1.3 as per Human Genome Project, ये कितना कर सकते हैं ये? They can code for 19 to 20,000 proteins by the most optimistic estimate. So most optimistic estimate सिर्फ इतनी protein synthesize हो सकती है. And by the most conservative estimate, 1 million immunoglobulins are synthesized. So how that is possible? On one side, we say only this much, और अगर सारे genes सिर्फ immunoglobulin भी ठीक करें, hypothetical, तब भी 1 million नहीं कर सकते. The problem is not as difficult, not as formidable as it appears to be, considering if we see the structure of the immunoglobulin. It's a tetramer comprising of 2 heavy chains and 2 light chains. So the antigen binding site is shown by this arrow. So antigen binding site, it comprises of 1 heavy chain and 1 light chain. Supposing if we have got 1000 types of light chains and same number of heavy chains, तो इनके कितने combinations possible है? 1000 into 1000 कितना हो गया? 10 raised to the power 6. So many combinations are possible. तो हमने 1 million को reduce कर दिया 2000. Only 2000 genes are sufficient to synthesize 1 million different types of antibodies, 1 million specificities. And these 2000 genes are easily created by gene rearrangement. Or in fact, we are going to see in a few minutes, not even 2000 are required. This is showing the antigen binding site in a more explicit manner. तो यहां पर क्या होता है, every, let us consider this light chain. It has got a variable region and a constant region. तो वेरिएबल रीजन और बीच में जॉइनिंग रीजन होता है. सो ओनली दिस पोर्शन वेरीज फ्रॉम वन अमाइनो एसिड टू अनदर. तो एक अगर लाइट चेन को अगर हम देखें, a light chain, it has got a variable region and it has got constant region. In between there is a joining region. और इनकी लेंथ वगैरह जस्ट एन आउटलाइन, जस्ट टू रिवाइज. 214 अमाइनो एसिड टोटल लाइट चेन में होते हैं. 440 are in the heavy chain. 108 of the light chain constitute the variable region. 118 of the heavy chain constitute the variable region of the heavy chain. So light chain made up of three regions: variable, joining, and constant, as we saw just now. Now, genes for these immunoglobulin light chain or the heavy chain, etc., they are on chromosome 2 and 22. There is more to that also, but let us stop at that. Now, these genes which are for these immunoglobulin chains, they comprise actually of gene segments or gene clusters, also called library of V genes. You will understand it in a few minutes. For variable region, there are approximately 48 genes. For constant region, a single gene codes for the constant region. For the joining region, there are about 6 genes. So these are actually gene segments. Okay, just very quickly I will show you, then I will return. Now for example, variable region का हमने बोला 48. Just concentrate on this, forget about this. So these are the gene segments, and here will be 48. Sure. We return to it. So the gene segments are there. V, J का 6, constant का 1. I am reading out again. Library of V genes code for variable region, number 48. Library of J genes codes for joining region, number 5 to 6, let us write 6, and a single gene codes for constant region. Now, one gene or one or two genes may be picked up at random from the variable gene segment. ये germline gene मैंने फिर दिखाए हैं. Let us say we pick up 8th and 9th gene from the variable region. 4th gene segment we pick up from the J region. तो उन दोनों को स्प्लाइस कर दिया. स्प्लाइस करने के बाद क्या बन गया? Complete variable region gene. All right, complete variable region gene. So I'm repeating because it is a bit mind-boggling. One or two V genes selected at random. One J gene select किया, splice to form a complete variable region gene. So it can be anything. It can be actually V11, V28, J2. तो इस तरह से कितने कॉम्बिनेशंस पॉसिबल है? जस्ट थिंक अबाउट इट. और दिस इज ट्रांसक्राइब अलोंग विद सी जीन. सी जीन तो सिंगल होता है, तो सिंगल मैसेंजर आ रहे हैं. So how many types of such combinations are possible? Come on, think about it. Innumerable, innumerable. So 1 million pales into insignificance when you consider all these points. All right. So 1 million gene by transposition can be easily synthesized. Now I am considering हैवी चेन, अभी हमने लाइट चेन देखा था, हैवी चेन के अंदर भी अप्रोक्सीमेटली सेम नंबर ऑफ joining joining genes and V genes are there. They have got an additional about D segment. D segment इसके अंदर भी I don't remember exactly कितने होते हैं, speak loudly, चलो जो भी है, I think 30 genes are there. Let's assume. I'll tell you the exact number in a few minutes. Dr. Adel will tell 23, 48, 23 and 6. तो कितने कॉम्बिनेशन पॉसिबल हो गए यहाँ पर V4D3J2, इस तरह से कंप्लीट वेरिएबल डोमेन जीन बन गया और उसको कांस्टेंट के साथ कर दिया और रिपीटिंग, मेरी एक्चुअली जबान लड़खड़ा रही है बिकॉज द नंबर इज माइंड बॉगलिंग. सो हमने जो शुरू में देखा था 10 raised to the power 11, that is nothing if you see all this. And there is something more to the story also. Various alleles for each and every gene may be present. जैसे number 3 genes हैं, so allele means different variants of the same gene. So allelic heterogeneity is very common. All right. So repeat ये final statement. Use of separate gene libraries or separate gene segment for assembly of the variable domain gene. हमने जैसे देखा था, ये जैसे चेन है कोई भी, तो ये वेरिएबल डोमेन होता है. This is the major mechanism for generation of antibody diversity. Okay, so this is, so one important thing, a gigantic problem in a stepwise manner, it has been reduced to very simple solution. Additional mechanisms are also there. High rate of somatic mutation in variable domain genes. Second, we saw that there is splicing or joining of various gene segments. There may be some imprecision overlapping of the joining of the gene segment. And then there may be enzyme addition of random nucleotides. And there are certain template independent DNA polymerases. So this is a bit complex. Think about it. And that is all about it. Antibody diversity. There were 3 more attributes of adaptive immune response: specificity, self, non-self discrimination, and memory. Think about it, try to answer yourself what forms basis of those 3 attributes also. So we conclude today, and Dr. Edelbert, he has been providing me with all the numerical figures, and wherever I get stuck, and for recording of this also, you will learn more about him in years to come. Thank you. Thanks a lot.